Welcome back to The Deep Dive, the show where we cut through the noise and get straight to the insights that make you truly well-informed. Today, we're embarking on a journey inside the human body itself, to the microscopic world that dictates almost every single thing we do.
That's right. We're taking a deep dive into the incredibly intricate world of hormone signaling. It's essentially how your body's cells and tissues communicate, sending vital messages from one place to another, and how those messages are received and acted upon. It's a system of remarkable elegance, really, turning simple molecular signals into profound changes in how your body functions.
And our mission today isn't just to skim the surface. We're going to unravel the complex language of the body, pulling out the crucial insights and surprising details from the foundational sources you've shared. We want you to walk away not just knowing what hormones do, but truly understanding how they do it, giving you a comprehensive grasp of this vital field. Let's jump in.
Okay, so at its most basic level, the fundamental purpose of hormones. It's really what makes multicellular life possible.
Right, because different parts need to coordinate somehow.
Exactly. Think about it. Once organisms evolved beyond single cells, different parts of their bodies needed a way to talk to each other, to coordinate actions. Hormones are that sophisticated messaging service. They carry vital information, maybe about what's happening in your environment or during development, and they transmit it across, vast distances within your body.
And the key step you mentioned is signal transduction.
Yes, signal transduction. It's a critical term. It's how a cell actually detects a hormone's presence, its concentration, and then converts that external signal into a specific internal action. And here's a cool fact. The communication strategies hormones use. They're often identical or very similar to those used by neurotransmitters or even drugs or basic metabolic compounds.
Interesting. So when we talk about classic endocrinology, what's the picture?
The classic picture is these signaling molecules, the hormones, hitching a ride on your bloodstream, traveling from where they're made, say, a gland, to a distant target tissue.
At that journey, it means they get diluted.
Yeah. Diluted, exactly, a lot.
Yeah.
So your target cells have to be incredibly sensitive. They've got to be able to pick up and respond to extremely tiny concentrations of these hormones.
And you mentioned the discovery process was almost like a detective story.
Oh, absolutely. For many of the classic hormones, it followed this stereotypical four-step process. First, researchers would see a disease-like syndrome after removing an endocrine gland.
Okay.
Second, the symptoms would vanish if they put that gland back in. Reimplantation.
Makes sense.
Third, they'd find they could get the same cure just by giving an extract from the organ.
Getting closer.
And finally, the fourth step. Purifying the actual active agent, the hormone itself. Insulin is the prototype here.
Right, like you said, insulin, thyroid hormone, cortisol, all discovered this way.
Exactly. And historically, we've kind of grouped hormones based on where their receptors are located, the little antennas picking up the signal.
Two main groups.
Yeah, basically two. First, you have those that bind to receptors right on the cell surface. These are often larger molecules, polypeptides like growth hormone or insulin, but also smaller ones like serotonin or prostaglandins.
And the second group?
The second group consists of hormones that are actually small enough or lipophilic enough to slip right inside the cell. They travel through the membrane and bind to receptors inside, often in the nucleus, directly influencing your genes.
Like steroid hormones.
Steroid hormones, thyroid hormones, those are the classic examples of intracellular access.
But you hinted it's not always that clear-cut.
No, it really isn't. As our tools get better, we see these definitions aren't as black and white. Many molecules are real multitaskers.
Like IGF-1.
Insulin-like growth factor 1, perfect example. Produced in the liver, circulates like a classic hormone, affecting growth body-wide. But it also has local paracrine effects, acting on nearby cells, like in bone development.
Or norepinephrine.
Right, neurotransmitters, nerve endings for quick local signals, but release it from the adrenal gland into the blood. Boom, it's a classic hormone acting far away.
Even testosterone isn't just nuclear.
Exactly. We know it binds nuclear receptors, but it also acts locally, in a paracrine way, on seminiferous tubules in the testes.
And cytokines, too. Usually inflammation, but they can act hormonally.
Yes. Cytokines released at inflammation sites can definitely circulate and act like hormones. Think about how some evoke fever by acting on the brain.
So it really pushes us to think beyond those older, stricter definitions.
It absolutely does. The body's systems are incredibly adaptable and interconnected.
Okay, so this brings up a basic challenge. How do the water-soluble hormones get their message across the cell membrane, that outer wall?
That's a fundamental point. The plasma membrane is this, 5 to 8 nanometer fluid mosaic, phospholipid heads facing out, fatty tails facing
A
in.
greasy barrier, basically.
Actually, yes, a non-polar environment. Great for organization, but it prevents water-soluble molecules from just diffusing across.
So that's where the receptors come in, the surface ones.
Precisely. Those surface proteins, the receptors, are absolutely essential. They're like specialized, secure mailboxes. They're designed to detect the right incoming ligand, the hormone, and then transmit that vital information across the membrane to kick off the internal signaling cascade.
And it's not just where the receptors are, but how the hormones are released. That seems incredibly tailored, too.
Oh, definitely. Think about peptide hormones like insulin. They're pre-made, stored in these tiny sacs, secretory vesicles, or granules, ready to go.
Which allows for fast release.
Incredibly rapid release, milliseconds, often. And their effects can shut off just as quickly because they typically have very short half-lives in the blood. Quick on, quick off. They bind receptors very quickly, too, what we call high-on rates. But interestingly, they often have slow off rates. They stick around on the receptor.
Which helps detect low levels, you said.
Paradoxically, yes. That slow dissociation helps the cell keep detecting the signal, even when hormone levels drop very low. And there are always exceptions, little surprises, like asparagine. It's a peptide, but it's not stored. It's cleaved off the cell surface when needed.
And IGF-1 is different, too.
IGF-1, yes. Unique among peptides because it circulates bound to binding proteins. This extends its lifetime dramatically.
So it influences longer-term things like growth.
Exactly. Growth, differentiation. Then you have smaller hydrophilic hormones, adrenaline, melatonin, serotonin, stored in even tinier, different-looking vesicles.
Okay, now compare that to the lipophilic ones, like steroids.
Totally different strategy. Steroids are generally not stored. They're synthesized on demand, and being lipid-soluble, they just diffuse right across cell membranes. No storage needed.
Thyroid hormone is another interesting case, though.
It is. It's a bit more hydrophilic than steroids. It's stored bound to a large protein, thyroglobulin, in the thyroid gland. And critically, it needs specific transporter proteins like MCT8 or OATP1 to actually get into target cells.
And problems with those transporters, like MCT8, cause serious issues.
Severe neurological problems, yes. It highlights how vital that transport step is. And remember, not all hydrophobic ligands sneak inside. Some still bind surface receptors, eicosanoids, for example. There are even GPCRs that respond directly to free fatty acids.
It's complex. And you also mentioned the relationship between ligands and receptors isn't always one-to-one.
Rarely is. It's often more intricate. One ligand, like norepinephrine, can bind to several different receptor types, miR1, MATE2, 3 adrenergic receptors, like one key opening multiple locks.
And the reverse. One receptor, multiple ligands.
That happens too. The mineralocorticoid receptor binds both aldosterone and cortisol. The androgen receptor binds both testosterone and its more potent derivative, DHT.
So with all this variety, how does the cell consistently get the right message? How does it avoid, confusion?
That's the crucial question. How does the body ensure fidelity? Well, for a target cell to generate the correct biological response when a hormone arrives, it needs 3 key things. First, specificity. The receptor has to distinguish this hormone from everything else floating around.
Makes sense.
Second, affinity. It must recognize and bind the hormone even at those incredibly low concentrations found in the blood. High sensitivity.
Okay.
And third, conversion. That initial binding event has to be effectively translated or converted into a specific cellular action. Recognition isn't enough. It has to trigger something.
And the strength of the effect relates to how many receptors are bound.
That's the fundamental rule, yes. The biologic effect is directly proportional to the fraction of receptors occupied by the ligand, which leads to this interesting concept of spare receptors.
Spare receptors, like having extras.
Exactly like having extras. Imagine you only need to flip a few light switches to get the room fully lit, even if there are many more switches available. The cell can achieve its maximal biological response by occupying only a fraction of the total available receptors.
So even with fewer receptors, you might still get a full effect.
You might, yes, but it might take a higher dose of the hormone to achieve it. The ED50, the effective dose for 50% response, would shift. Insulin is a classic example where this happens.
But we can actually measure this in the lab. Specificity, affinity.
Oh, yes. Using radioactively labeled ligands is key. We can measure how tightly they bind, affinity, how specific that binding is, and at what point the receptors become saturated, meaning they're all occupied.
Okay, so hormones bind, often tightly. How does the signal eventually stop? Diffusion isn't enough.
Not usually, especially with those slow off rates. The body needs active off switches. A major one is receptor-mediated endocytosis.
The cell swallows the receptor.
Basically, yes. The whole ligand-receptor complex gets internalized, then it's either destroyed or the receptor might be recycled back to the surface.
What else stops the signal?
There are specialized internal desensitizer proteins, things like arrestins, which we'll talk more about, and SOCS proteins. They can physically block the receptor from signaling further.
And phosphorylation, adding or removing phosphate tags.
Absolutely critical. Elegant on-off switches. Adding phosphates, phosphorylation, or removing them, dephosphorylation, at specific sites on the receptor can dramatically change its activity. The insulin receptor uses this extensively.
Okay, so given all this, how are hormone levels actually kept in check? It sounds like tight regulation is essential.
Incredibly tightly controlled. It's like a finely tuned orchestra. There are multiple mechanisms at play. First, precursor availability. Simple enough. No raw materials, no product. Iodine deficiency causing hypothyroidism is the classic example. Second, the actual synthesis and secretion are regulated, often by hormones from the pituitary gland, like TSH controlling thyroid hormone production. If the gland itself fails, you get deficiency.
What about activating hormones?
That's third, activation. Many hormones start as inactive precursors, or prohormones, like pro-opiomelanocortin for ACTH or pro-insulin for insulin. They need specific enzyme, prohormone convertases, to snip them into the active form. Defects in these enzymes cause disease.
And hormones can be changed after release, too.
Yes. Fourth is biotransformation. This can happen in the target cell or elsewhere. Think of T4 being converted to the more active T3. Or testosterone to estrogen via aromatase. Vitamin D activation is another key example. Testosterone to DHT via 5-reductase.
And transport into the cell can be regulated.
Number 5. Transport into the target cell. We mentioned the thyroid hormone transporters MCT8 and OETP1. Mutations there prove this step is vital. Steroids mostly diffuse, but for others it's a regulated gate.
And the receptor's own sensitivity plays a role in feedback.
Number 6. Receptor signaling and sensitivity. Absolutely. If receptors become less sensitive, like in insulin resistance, the body often compensates by cranking up hormone production. The pancreas makes more insulin. Leptin from fat tissue signaling energy stores to the brain is another great example of this feedback.
And finally, getting rid of them.
7th, elimination. Crucial for turning signals off. This happens via liver or kidney clearance or specific enzymes breaking them down, like deiodinases inactivating thyroid hormone. Problems with inactivating enzymes can cause hormone excess symptoms. And for large protein hormones, receptor-mediated endocytosis is also a major elimination pathway.
Okay, that's a fantastic overview of the whole system. Shall we dive into the receptors themselves, starting with the surface ones?
Let's do it. First up, the largest family. G, protein-coupled receptors, or GPCRs, the 7 helix wonders.
7 helices. So they snake through the membrane 7 times.
Exactly. That's their structural hallmark. The N-terminus, the beginning of the protein chain, is outside the cell. The C-terminus is inside. They all rely on these helpers called heterotrimeric G proteins.
And there are lots of them.
Over 800 in humans. Though, interestingly, the vast majority are actually olfactory receptors involved in your sense of smell.
But the ones involved in hormone signaling detect a huge range of things.
An incredible range. Single photons of light, ions, tiny odor molecules, amines like adrenaline, peptides, lipids, nucleotides, metabolic products, even large proteins. They are remarkably versatile.
Where does the hormone actually bind?
It depends. Smaller hormones often bind down within that transmembrane helix bundle. Larger peptide or protein hormones usually bind to the extracellular N-terminus or loops sticking out of the cell.
And they're grouped into families.
5 main subfamilies based on their sequences. You've got glutamate, rhodopsin-like, which includes receptors for many hypothalamic hormones, glycoprotein hormones, amines adhesion, frizzltase 2, and secretin-like, which bind things like glucagon and PTH.
You mentioned orphan receptors, too.
Yes, these are GPCRs where we haven't yet identified the natural ligand, the molecule that activates them. Some might even be constitutively active, always sending a low-level signal. Still a lot to discover there.
So how do these G proteins actually work? You call them timed switches.
That's a good analogy. These G proteins are made of 3 parts: an alpha, a beta, and a gamma subunit. Normally, the alpha subunit holds onto a molecule called GDP, which keeps it inactive.
It's like a dead battery.
Sort of, yes. When the hormone binds the GPCR, the receptor changes shape and interacts with the G protein. This interaction causes the alpha subunit to release that GDP. And because there's much more GTP inside the cell, GTP binds instead. That's the fresh battery.
And the GTP binding activates it.
It activates the alpha subunit and, at least in test tubes, causes it to dissociate from the beta-gamma dimer. Both the GTP-bound alpha subunit and the free beta-gamma dimer can then go off and interact with downstream targets like enzymes or ion channels propagating the signal.
So the alpha part activates things, and the beta-gamma part can too.
Absolutely. GS-alpha activates adenyl cyclase, for instance, but beta-gamma dimers can activate potassium channels, among other things.
And how does a timer work? How does it switch off?
The alpha subunit has a slow, built-in ability to break down GTP back to GDP. It's a GDPase. Once it's GDP bound again, it becomes inactive and rejoins the beta-gamma dimer.
But you mentioned RGS proteins.
Right. That intrinsic GTPase activity is often too slow. So regulators of G protein signaling, RGS proteins, act as GTPase accelerating proteins, or GAPs. They speed up the GTP hydrolysis significantly, shortening the duration of the signal.
So RGS levels are a control point.
A key control point. For example, TSH and PTH can actually increase the expression of RGS2, which then feeds back to inhibit the very G protein Gs that they activated. It's a negative feedback loop.
Okay, beyond G proteins, you mentioned arrestins as another crucial regulator.
Yes, arrestins. There are 4 types, 2 mainly in vision, and 2, rho-arrestin 1 and 2, that are found everywhere. Often, when a ligand binds a GPCR, it triggers phosphorylation of the receptor itself by a specific kinase family called G protein receptor kinases, GRKs.
Adding those phosphate tags again.
Exactly. Those phosphate tags act as a signal for arrestin to bind. When arrestin binds, it physically blocks the GPCR from interacting with the G protein anymore. It effectively uncouples them, terminating that G protein signal.
Pulling the plug, like you said.
Right. But arrestin does more. Binding also changes arrestin's shape, allowing it to interact with components of the cell's endocytosis machinery, like clathrin. This leads to the GPCR being internalized, pulled into the cell for degradation or recycling. This is called homologous desensitization because it specifically affects the activated receptor type.
And arrestins can signal themselves.
That's a fascinating development. Yes, arrestins can act as signaling scaffolds, bringing together other kinases and signaling molecules like GRC, MAPKs, PI3K, ACT, initiating entirely different downstream pathways. They're not just off switches.
Which leads to this idea of biased signaling.
Precisely. Bias signaling. It's the idea that different ligands binding to the same GPCR, or even the same ligand under different conditions, can stabilize slightly different receptor conformations.
Different shapes.
Different shapes, yes. And these different shapes can preferentially activate one pathway over another, maybe favoring one type of G protein, or maybe favoring G protein signaling while minimizing arrestin recruitment and internalization.
And that has huge pharmaceutical implications, like for opioids.
Huge. The holy grail for opioid development is finding a ligand that strongly activates the G protein pathway for pain relief, analgesia, but weakly activates or avoids activating the arrestin pathway, which seems linked to tolerance, constipation, and respiratory depression.
So bias can be influenced by lots of things. Ligand type, concentration.
Ligand type, concentration, which GRKs are available to phosphorylate the receptor where the receptor is located in the cell, even the timing of the signal. It's incredibly nuanced.
Given this complexity, it makes sense that mutations can cause disease.
Definitely. A number of endocrine diseases result from mutations in GPCRs or G proteins.
Often loss of function.
Often, yes, leading to hormone resistance. Mutations in the TRH or TSH receptors causing hypothyroidism, or the ACTH receptor causing glucocorticoid deficiency. Melanocortin-4 receptor mutations leading to extreme obesity. These are often recessive.
But gain of function happens too. Receptor stuck on.
It does. Activating mutations in the TSH receptor cause non-autoimmune hyperthyroidism. Mutations in the calcium-sensing receptor can cause hypoparathyroidism. Sometimes these are dominant.
And G protein mutations, mostly the alpha subunit.
Primarily, yes. A mutation in gut transducin causes night blindness. Dominant activating mutations in GUS are found in many pituitary adenomas, especially GH-secreting ones.
And Albright hereditary osteodystrophy. That involves GUS and imprinting.
Yes, AHO. It's caused by loss of a functional GUS allele. Whether you also get thermal hypoparathyroidism type 1A depends on whether you inherit the defective allele from your mother or father due to genomic imprinting, where only one parent's copy is normally active in certain tissues. Fascinating genetics.
Okay, let's shift gears to the next major class. Receptor tyrosine protein kinases, or RTKs, the phosphorylators.
Right. These are different because the receptor itself has intrinsic enzyme activity, specifically tyrosine kinase activity.
So hormone binds, enzyme switches on inside.
Exactly. They are all type I transmembrane proteins. An N-terminal hormone binding domain outside, a single pass through the membrane, and a C-terminal portion inside that contains the kinase domain. This intracellular domain transfers phosphate from ATP onto tyrosine residues on target proteins, including the receptor itself that's called autophosphorylation.
And there are quite a few of these.
Around 58 in humans, grouped into about 20 subfamilies.
The insulin receptor is a bit unusual in this group, isn't it?
It is unique. Most RTKs exist as monomers that dimerize upon activation, but the insulin receptor is already a preformed disulfide-linked tetramer, 2 alpha subunits, 2 beta subunits, even in the absence of insulin.
Made as one piece, then cut.
Synthesized as a single peptide, then cleaved into the alpha and beta chains in the Golgi apparatus. And insulin binding shows negative cooperativity.
Yes. Binding in the first insulin molecule actually decreases the receptor's affinity for binding a second insulin molecule. It has 4 potential binding sites, but one insulin effectively cross-links the two halves, making it harder for the second high-affinity site to bind another insulin.
Other RTKs activate differently. Yes, the EGF receptor, for example, involves one EGF molecule binding to one receptor monomer, which then pairs up with another. PDGF binds as a dimer, bringing 2 receptor monomers together.
But the general theme for activation is dimerization, bringing 2 receptors together.
Generally, yes. Activation requires receptor dimerization or a change in the arrangement of a preformed dimer.
But if some are already dimers, like the insulin receptor, dimerization itself isn't the trigger.
Correct. For preformed dimers like insulin, IGF-1, FGF, EGF receptors, something more fundamental has to change in their interaction upon ligand binding.
For insulin IGF-1, you described an inverted V closing.
Yes. The unbound receptor's extracellular domains are like an open V. Ligand binding seems to release a brake on a molecular hinge, causing the V to close, bringing the parts spanning the membrane closer together.
And that change gets transmitted inside.
It does. It brings the 2 intracellular kinase domains into proximity. Now, in the inactive state, a part of the kinase called the activation loop physically blocks the enzyme's active site, but proximity allows the kinase domain of one beta subunit to reach over and phosphorylate tyrosines in the activation loop of its partner subunit. This is transphosphorylation.
And that phosphorylation activates it.
It forces the activation loop to move out of the way, opening up the catalytic site and activating the kinase. This activated kinase then phosphorylates its partner, fully activating both.
Is the EGF receptor different?
Yes, its activation is thought to be more about allosteric regulation. One kinase domain physically interacts with and activates the other simply by being pushed right up against it. Proximity drives it.
So once these RTKs are active, how do they signal downstream?
Primarily by recruiting a variety of signaling proteins. The autophosphorylation creates docking sites.
Phosphorylated tyrosines as docking sites.
Exactly. Specific phosphorylated tyrosines serve as binding sites for proteins containing SARS-2 homology 2, SH2 domains, or phosphoryracine binding PTB domains. These domains recognize the phosphorylated tyrosine within a specific amino acid sequence context.
Like the PI3K pathway.
A key example, the PI3K enzyme, which has a regulatory subunit with SH2 domains, gets recruited to a specific phosphorylated tyrosine on the activated receptor. This recruitment does 2 things. It activates PI3K and, crucially, brings it right next to its substrate, a lipid called PI4005P2, which resides in the plasma membrane.
Okay.
PI3K then phosphorylates PI4005P2 to create PIP3, phosphatidylinositol 3004-4-maltose phosphate. PIP3 is itself a potent signaling molecule that recruits other proteins to the membrane via their pH domains.
So it's about spatial relationships,
Right.
bringing things together.
That's a core principle of RTK signaling, establishing adjacency between critical signaling molecules and their targets or activators.
What about activating rays? How does that work?
RAS activation often starts with recruitment of adapter proteins like SHC or GRB2 via their SH2 or PTB domains. SHC might get phosphorylated by the RTK first, allowing GRB2 to bind. GRB2 is constitutively bound to another protein called SOS, son of sevenless. So recruiting GRB2 effectively drags SOS to the plasma membrane.
And SOS activates RAS.
SOS is a guanine nucleotide exchange factor, GEF, for RAS. Its job is to catalyze the removal of GDP, the off signal from RAS, and allow GTP, the on signal, to bind. The critical event is positioning SOS right next to RAS at the membrane.
Now, insulin and IGF-1 receptors use a variation involving IRS proteins.
Yes, they predominantly use insulin receptor substrate, IRS proteins, mainly IRS-1 and IRS-2, as intermediary docking platforms.
IRS proteins are just scaffolds, no enzyme activity.
Correct, they are docking proteins. The activated insulin receptor heavily phosphorylates IRS-1 and IRS-2 on multiple tyrosine residues. This creates numerous binding sites for the SH2 domains of downstream players like PI3K, GRB2, and others, assembling large signaling complexes on the IRS protein.
How do IRS proteins get to the receptor?
They have N-terminal domains, pH and PTB, that help target them to the activated receptor near the membrane. While this tyrosine phosphorylation happens rapidly to turn the signal on, there's also slower serotonin phosphorylation of IRS proteins, often mediated by downstream kinases. This serves as negative feedback, inhibiting further tyrosine phosphorylation or even marking IRS for degradation.
Are there other scaffolds involved with insulin?
Yes, SHC can also be recruited and phosphorylated, leading to RAUS activation via the GRB2 SOS route, and there's GRB10. An inhibitor. GRB10 is interesting. It's an SH2-containing protein that binds to phosphorylated tyrosine specifically in the activation loop of the insulin receptor. This binding actually blocks the receptor's kinase activity, inhibiting PIP3 production. It acts as a negative regulator. Disruption of GRB10 in mice leads to overgrowth.
What about SH2B1 and SH2B2?
They also bind the phosphorylated insulin receptor. SH2B1 seems to enhance insulin action. Its deficiency decreases sensitivity. SH2B2 might negatively regulate sensitivity as its deficiency seems to increase it, so more fine-tuning. All this complexity naturally leads to thinking about insulin resistance syndromes.
When the system breaks down.
Exactly. Inherited forms caused by mutations in the insulin receptor gene itself lead to a constellation of symptoms: hyperinsulinism, metabolic problems, a skin condition called acanthosis nigricans, hypomandrogenism in females.
And severe mutations cause leprechaunism.
Very severe loss-of-function mutations, yes, leading to profound developmental defects. Mutations can decrease receptor number, impair insulin binding, or cripple the kinase activity. This genetic form is sometimes called type A resistance.
How about type B resistance?
Type B often presents later in life and is frequently associated with autoimmunity conditions like vitiligo, alopecia, arthritis. It's caused by the body producing antibodies against the insulin receptor.
But the common insulin resistance seen in obesity or PCOS, that's different.
Yeah.
It's often different, yes. Sometimes calling it just insulin resistance can be a bit misleading when comparing it to the severe genetic forms. In obesity-related resistance, while glucose metabolism is impaired, some other insulin actions might be preserved. It doesn't fully mimic the phenotype of having a completely broken receptor.
So it's more selective, a post-receptor defect.
Often, yes. The idea is that factors associated with obesity, like chronic inflammation or elevated free fatty acids, induce selective insulin resistance by interfering with specific downstream signaling pathways, rather than causing a global defect in the receptor itself.
Okay, shifting again. What about receptors that are serotonin kinases? The TGF receptors.
Right, a distinct mechanism. These receptors have intrinsic serotonin protein kinase activity, targeting different amino acids than the RTKs. Their ligands are members of the large transforming growth factor beta, TGFA family. Over 40 human agonists, including TGF itself, activins, inhibins, bone morphogenetic proteins, BMPs, myostatin, anti-Mullerian hormone.
All dimer.
Each ligand is a dimer, yes. Historically, inhibiting was found first made in the gonads, blocks FSH release, then activin a homodimer promotes FSH release.
And the receptors, also complexes.
Yes, the functional receptor is composed of 4 transmembrane glycoproteins, 2 type 1 receptors, and 2 type 2 receptors. A key structural difference is that type 1 receptors have a conserved region called the GS domain, which binds another protein called FKBP12 in the inactive state.
How does activation work here?
Typically, a ligand like activin first interacts with the type 2 receptors. This interaction then recruits the type 1 receptors, bringing them into close proximity. The type 2 receptors, which are constitutively active kinases, then phosphorylate that GS domain on the partner type 1 receptor.
And that phosphorylation is the switch.
It is. It relieves steric hindrance, causes FKBP12 to dissociate, and activates the type 1 receptor's kinase domain, allowing it to phosphorylate its downstream targets.
How does inhibin block this?
Inhibin uses a clever trick. It recruits another membrane protein called beta-glycan to form a stable complex with the type 2 receptors, essentially sequestering them and preventing them from activating their type 1 partners.
And the main downstream targets are SMAD proteins.
Yes, the primary intracellular signaling pathway involves the SMA proteins. There are 8 human SMAD genes.
Different types of SMAs.
3 main types. Receptor-regulated SMADs are SMADs, like SMADs 1, 2, 3, 5, 89. These have a specific site that gets phosphorylated directly by the activated type I receptors. Then, 2 phosphorylated RSMAs typically form a trimer complex with the common partner SMADG, which is SMA4.
And this complex goes to the nucleus.
Exactly. It translocates to the nucleus where it binds DNA and interacts with other transcription factors to regulate gene expression. SMAs themselves can get further modified in the nucleus, fine-tuning their activity. What about inhibitory SMAs? The third type, inhibitory SMAs 6 and 7. These act as negative feedback. They can bind to the activated receptor complex, promoting its ubiquitination and degradation, thus shutting down the signal.
You mentioned myostatin earlier, part of this family, the muscle growth regulator.
Yes, myostatin, or GDF8, a really interesting member. Secreted by skeletal muscle, acts as a brake on muscle growth. It binds to type 2 and type I receptors, leading to phosphorylation of SMA2 and SMA3.
And deficiency has dramatic effects.
Dramatic. It's responsible for the double-muscled phenotype in certain cattle breeds like Belgian Blue. In mice and even humans, mutations causing myostatin deficiency lead to massive muscle hypertrophy and hyperplasia, a clear demonstration of its role.
Fascinating. Okay, next group. Cytokine receptors and the JAK-STAT pathway. These receptors don't have their own enzyme activity.
Correct. They are enzyme associates. These receptors possess no intrinsic catalytic activity themselves. Instead, they associate non-covalently with separate cytoplasmic tyrosine kinases to transmit their signal.
Examples include receptors for GH, prolactin, leptin.
Yes, receptors for classic hormones like growth hormone, GH, prolactin, and leptin fall into this category, class I cytokine receptors, along with receptors for many interleukins and interferons. They are typically type 1 transmembrane proteins.
Activation involves dimerization again.
Often, yes. Dimerization or sometimes higher-order oligomerization. A single ligand molecule, like GH, can have 2 distinct binding sites, allowing it to bridge 2 receptor monomers.
Some are homodimers, some are heterodimers.
Right. GH, prolactin, leptin receptors are homodimers, 2 identical subunits. Others, like the IL-2 or IL-6 receptors, consist of a unique ligand-binding subunit plus 1 or more shared signaling chains, like MANGOC or GP130.
And again, some exist as preformed dimers, so binding causes a shape change.
Exactly. Many, including APO, GH, and prolactin receptors, seem to exist as dimers even before the ligand arrives. So ligand binding must induce a conformational change that reorients the intracellular domains.
And that understanding led to the drug pegvisomant.
Yes, pegvisomant is a GH antagonist used to treat acromegaly. It competes with natural GH for binding, but is designed so it cannot induce that functional conformational change needed for signaling. It prevents proper activation.
So these receptors associate with JK kinase.
Right next to the membrane, on the inside, these receptors have sequences critical for binding members of the Janus kinase JAK family of tyrosine kinases.
4 members, JAK1, 2, 3, and TYK2.
Correct. Homodimers, like the GH receptor, predominantly associate with JAK2. Heterodimeric receptors often bind multiple JK. For example, IF-NO receptor binds JAK1 and JAK2.
How do JAKs bind, and what's their structure?
They associate via a conserved N-terminal region. Structurally, they have a kinase domain that does the phosphorylating, but also, interestingly, a pseudokinase domain right next to it, which actually suppresses the activity of the kinase domain in the inactive state.
So activation involves relieving that suppression.
Precisely. For the GH receptor, ligand binding causes a conformational change outside, which translates into an intracellular scissors opening motion. This physically separates the pseudokinase domain from the kinase domain on the associated JAKs, relieving the inhibition and switching the kinase on.
Clever mechanism. So what happens once JAK is active?
The major immediate consequence is transphosphorylation. The activated JAK phosphorylates tyrosine residues on its partner JAK and critically on the cytoplasmic tail of the receptor subunits themselves.
Creating docking sites again.
Creating docking sites again, exactly. For SH2 or PTB domain containing signaling molecules, this can recruit pathways involving IRS proteins or PLSO.
But the key pathway for growth here is STAT.
Yes, perhaps more critical, especially for growth and differentiation signals, is the recruitment and activation of the signal transducers and activators of transcription STAT family.
7 STAT proteins. GH uses STAT-E5B.
GH receptor binds several, but STAT5B seems particularly critical for its growth-promoting actions. There are 7 STAT proteins in mammals, all sharing similar domain structures, including a crucial SH2 domain.
How does STAT5B get activated?
It gets recruited to those newly phosphorylated tyrosines on the receptor created by JK2 activity. Once docked, SECI5B itself becomes a substrate and gets phosphorylated on a key tyrosine residue, presumably by the receptor-associated JAKs.
And phosphorylation causes dimerization.
Yes, the phosphorylated tyrosine on one STAT binds to the SH2 domain of another STAT, forming a dimer. This dimerization causes the STAT complex to dissociate from the receptor.
Then straight to the nucleus.
It translocates into the nucleus, binds to specific DNA sequences, and directly regulates the transcription of target genes. It's a very direct pathway from receptor activation to gene regulation.
Any other layers of regulation?
Oh, yes. STAT activity can be further modulated by phosphorylation on serine residues by other kinases like MEKs. Proteins like SH2B1 can enhance JK2 activity. STAT proteins can also form heterodimers with other STATs or even cooperate with other transcription factors, like STAT5B working with the glucocorticoid receptor to activate the IGF-1 gene.
Leptin signaling is another important JAK-STAT example.
Absolutely. Leptin, from fat cells, acts on the hypothalamus to suppress appetite and increase metabolism. Deficiency causes massive obesity.
Same mechanism. Binds receptor, activates JK2.
Fundamentally, yes. Leptin binds to homodimers of its receptor, a class A cytokine receptor, activating associated JK2. There are several isoforms of the leptin receptor, but the long form, LEPARB, is the main signaling one. It primarily recruits and activates STAT3. It can also recruit STAT5 and a phosphatase called SHP2, which surprisingly acts positively here to activate the ERK and EPK pathway.
What turns leptin signaling off?
A phosphatase called PTP1B dephosphorylates both the leptin receptor and JK2, inhibiting the signal. Deleting PTP1B in mice actually protects them from obesity and insulin resistance.
And STAT3 is proving critical for leptin's effects.
Absolutely critical. There's a specific tyrosine, Y0138, on LEPARB required for STAT3 binding. Mutating that single amino acid blocks STAT3 recruitment and causes obesity almost as severe as having no leptin receptor at all. Clear proof of STAT3's role in appetite and energy balance.
How are these cytokine signals terminated more generally?
Several ways. Dephosphorylation of key tyrosines by phosphatases like PTP1B or SHP phosphatases is one. Another major mechanism is the transcriptional induction of suppressors of cytokine signaling, SOCS proteins.
Induced by STATs themselves. Negative feedback.
Exactly. SOCS genes are direct targets of STAT transcription factors, so the signal activates its own inhibitor. Very elegant feedback. SOCS proteins, there are 8 family members, have an SH2 domain to bind phosphorylated tyrosines on the receptor, or JAKs. They can directly inhibit JAK activity, compete with STATs for binding sites, and even target the receptor complex for degradation via the ubiquitin pathway, thanks to their SOCS box domain.
So SESOS2 deficiency causing gigantism makes sense.
Perfect sense. Enhanced GH signaling because the brake is faulty. There's also the protein inhibitor of activated STAT, PIS family, which inhibits STAT signaling by blocking DNA binding, recruiting corepressors, or promoting sammoylation.
So defects in these pathways explain certain growth disorders.
Yes, defects in cell surface receptors that control growth are well documented. GH resistance leading to short stature is the classic example. Lorne syndrome is the prototype high GH, low IGF-1, caused by diverse mutations in the GH receptor gene.
Similar syndromes from STAT5B mutations or IGF-1 signaling defects.
Exactly. Mutations in STAT5B cause a very similar phenotype. And of course, defects further downstream in IGF-1 signaling also impair growth.
What about short syndrome? That involves PI3K.
Yes, short syndrome is caused by mutations in the PIK3R1 gene, which encodes a regulatory subunit of PI3K. It results in a complex phenotype, including short stature, specific facial features, lipodystrophy, and also insulin resistance, again, highlighting the links between growth and metabolic pathways.
Okay, one last group of surface receptors, ligand-gated ion channels, rapid responders.
These are the simplest, in a way. The protein that binds the hormone is the channel, binding and signal generation in one package.
Structure. Ligand binding outside, channel through the membrane.
That's it. A ligand-binding domain on the cell surface and a transmembrane domain that forms the ion pore. Ligand binding causes a rapid conformational change that opens the pore, allowing specific ions like sodium, potassium, chloride, or calcium to flow across the membrane down their electrochemical gradient.
The classic example is the acetylcholine receptor.
The nicotinic acetylcholine receptor at the neuromuscular junction is the prototype. Acetylcholine binds, the channel opens, sodium rushes in, depolarizing the muscle cell and triggering contraction. Extremely fast, microsecond to millisecond timescale.
So mostly neurotransmitter receptors.
Primarily, yes. This family includes receptors for serotonin, 5-HT3 type, glycine, and inhibitory GABA type A receptors. They share a structural feature called a cis loop.
But some hormones use them. Serotonin regulating prolactin.
Yes, there are exceptions where classic hormones or releasing factors use these. Serotonin released into the pituitary portal blood can regulate prolactin release from lactotrophs via the 5-HT3 receptor, which is a ligand-gated cation channel.
Interesting. So some overlap even here.
Always overlaps in complexities in biology. Glycine and GABA receptors are also found in the pituitary, though their roles are less clear. And there are purinergic receptors responding to extracellular ATP that likely play local signaling roles there too.
Okay, that covers the surface receptors brilliantly. Shall we venture inside the cell now, to the nucleus?
Let's do it. Into the nuclear command center, receptors inside the cell. We're now diving into the deepest part of the cell's communication network, the nucleus itself. These receptors, the nuclear hormone receptors, are the true maestros inside. They are primarily known as ligand-activated transcription factors, meaning a hormone binds and they directly influence which genes get turned on or off.
Directly affecting DNA expression.
Exactly. Though, as we're increasingly realizing, they also have emerging non-genomic actions working outside the nucleus, too. More on that later.
It's a large family.
It's a superfamily, yes. 48 members in humans. Found in all multicellular animals, critically important for coordinating complex life, but absent in single-celled organisms like yeast.
And about 12 bind classic hormones. Steroids, vitamin D, thyroid hormone.
Right, those are the well-known ones.
Okay.
But that leaves 36 that were originally orphan receptors. We didn't know they're ligands.
Have we found ligands for them?
For many, yes. About 26 are now considered adopted orphan receptors with identified ligands, often lipids, metabolic intermediates, or bile acids. And studies show many of these are absolutely essential for life or proper organ development.
Some might be always active.
Some orphan receptors appear to be constitutively active, influencing gene expression even without a known external ligand. Still areas of active research.
What's the general structure of these nuclear receptors?
They are typically proteins of about 5,100 kiloday, sharing a common modular domain structure, usually labeled A through F.
Okay, break that down. AB domain first.
The AB domain is at the N-terminus, the beginning. It's the most variable in size and sequence between different receptors. Its function is least defined, but it often contains an activation function 1, AF1.
AF1, what's that?
It provides ligand-independent positive transcriptional activity. Its importance varies. It seems more critical for steroid hormone receptors, which tend to have longer AB domains and can contribute to tissue-specific actions.
In the C domain, DNA binding.
Exactly. The C domain is the highly conserved DNA binding domain, DBD. About 66 to 68 amino acids, forming 2 zinc finger structures. This is what recognizes and binds to specific DNA sequences called hormone response elements, HREs.
D domain.
The D domain is often considered a hinge region, providing flexibility between the DVD and the next major domain.
Which is the E domain, a lesion binding.
Yes, the E domain is the C-terminal ligand binding domain, LDD. This is where the lipophilic hormone binds with high affinity. But the LBD does more. It's also crucial for receptor dimerization and interaction with co-regulator proteins that modulate transcription.
And the F domain, still a mystery.
Largely, yes. The F domain at the very C-terminus is hypervariable in length and sequence, and its specific function remains unclear for most receptors.
So these are made in the cytoplasm, but work in the nucleus. How do they get there?
They're synthesized on cytoplasmic ribosomes, like other proteins. Then they are imported into the nucleus. This is directed by a nuclear localization signal, NLS, usually located near the border between the C, DBD, and D hinge domains.
And most just stay in the nucleus.
Most nuclear receptors reside predominantly in the nucleus, whether or not their specific hormone ligand is present. They might be bound to DNA already.
But the glucocorticoid receptor is the exception.
It's the classic exception, yes. In the absence of hormone, the glucocorticoid receptor is tethered in the cytoplasm, bound to a complex of chaperone proteins like heat shock proteins, HSPs. Hormone binding triggers a conformational change, causing the chaperones to dissociate. This unmasks the NLS, allowing the receptor to translocate into the nucleus.
Let's talk about ligand binding. How is it so precise?
The exquisite precision of ligand binding is down to the LBD's structure. It folds into a complex tertiary structure, typically involving about 12 alpha helical segments.
Forming a pocket.
Exactly. A hydrophobic pocket is formed within this structure, primarily by helices 3, 4, and 5. The specific shape and chemical nature of this pocket determines which ligand fits snugly.
And binding causes a big shape change. The cap.
A major structural change occurs upon ligand binding. The most C-terminal helix, often called H12 or the AF2 helix, folds inward, acting like a lid or cap on the ligand binding pocket, trapping the hormone inside. The precise way H12 repositions is critical for subsequent steps like co-regulator binding and dictates ligand specificity.
Okay, so the receptor binds its hormone. How does it find the right genes, HREs?
Target gene recognition by receptors relies on that DNA binding domain, CE domain, recognizing and binding to specific hormone response elements, HREs, which are short DNA sequences usually located near the genes the receptor regulates.
What determines which HRE a receptor binds?
Specificity comes from several factors. Within the DBD, a region called the P box contains amino acids that make direct contact with the DNA bases of the HRE half site. This sequence largely determines whether a receptor recognizes the steroid receptor half site, like AJAKA, or the half site used by most others, AGTCA.
Half sites, because they bind as pairs.
Yes, they typically bind as dimers, recognizing two HRE half sites. The spacing and orientation of these half sites are also absolutely crucial for specificity.
Let's talk about that dimerization dance. Steroid receptors first.
Steroid receptors, like the estrogen, progesterone, androgen, and glucocorticoid receptors, function primarily as homodimers, two identical receptors pairing up. They generally prefer HREs arranged as inverted repeats, palindromes, typically with a specific spacing of 3 base pairs between the half sites, an IR3 element. Dimerization involved contacts in both the DBD and LBD, and ligand binding facilitates this process.
And the others. Thyroid hormone, vitamin D, PPRs.
Right.
They partner with RXR.
Yes, most other nuclear receptors, including those for thyroid hormone, TR, retinoids, RXR is like a common partner.
Exactly.
Yeah.
Heterodimerization with RXR is mediated by strong interactions, especially between the LBDs, even without DNA, and also DBD interactions that are sensitive to the HRE structure.
And these heterodimers prefer direct repeats with specific spacing. Right. They typically bind to HREs arranged as direct repeats, DRs, of the AGDCA half site, and the number of spacing nucleotides between the repeats is a major determinant of specificity.
Like a code? DR3 for VDR, DR4 for TR, DR5 for R?
Precisely. VDRXR prefers DR3, TRXR prefers DR4, RARXR prefers DR5. RXR typically binds to the upstream half site. The way the DNA helix twists also influences how the DBDs interact across these different spacings.
So the combination of the receptor pair and the HRE spacing dictates which genes are targeted.
That's a key part of the specificity. And while we used to find these sites empirically, modern techniques like ChIP-seq allow us to map all the binding sites for a given receptor across the entire genome in a specific cell type. This is called the cistrome.
And the cistrome varies by cell type because of pioneer factors.
Exactly. The cistrome is highly context-dependent. A receptor might have tens of thousands of potential HREs in the genome, but it only binds to a subset in any given cell. This is largely determined by the chromatin state whether the DNA is accessible. Pioneer transcription factors are often responsible for initially opening up tightly compacted chromatin, heterochromatin, at specific locations, creating landing pads that allow the nuclear receptors to then bind their HREs.
So they bind DNA. How do they actually regulate the genes? Orchestrating gene transcription.
They mediate diverse effects. The most common is ligand-dependent activation. Hormone binds, receptor turns the gene on more strongly, but they can also mediate ligand-independent repression. Receptor sits on DNA without hormone and turns the gene OFF. And sometimes ligand-dependent negative regulation or transrepression, hormone binds, receptor turns the gene OF.
And co-regulators are key to this, coactivators and corepressors.
Absolutely essential. Much of this regulation involves the recruitment of these large protein complexes.
Let's start with activation. Hormone binds, gene transcription increases.
Right. Ligand-dependent activation. The DBD brings the receptor's activation domains close to the gene's start site. A key activation domain is within the LBD, called activation function 2. AF2, whose structure is critically dependent on that H12 helix position we discussed.
How does it boost transcription? Recruiting coactivators.
Precisely. The ligand-bound conformation of the LBD, particularly the surface created by H3, H4, H5, and the repositioned H12, creates a specific binding site for coactivators.
What do coactivators do?
They bridge the receptor to the general transcription machinery, like RNA polymerase II, and very importantly, modify chromatin structure. Many coactivators possess or recruit histone acetyltransferase, HAT activity.
Adding acetyl tags to histones, loosening the DNA.
Exactly. Acetylation neutralizes positive charges on histones, loosening their grip on the negatively charged DNA, making the gene more accessible for transcription. Think of it as opening up the chromatin. Key coactivator families, like the P160s, for example SRCs, have HAT activity and also recruit other HATs like CBPP300 and PCF.
Other mechanisms too. DNA unwinding, mediator complex.
Yes, some coactivator complexes contain chromoneme remodeling engines, like SWE-SNF, that use ATP to physically reposition or eject nucleosomes. And the large mediator complex acts as a central hub, physically linking the DNA-bound receptors and coactivators to the RNA polymerase machinery. And this is dynamic, cycling on and off, highly dynamic. Coactivators and even the receptors themselves cycle on and off the promoter region over timescales of minutes, suggesting transcription occurs in bursts.
Okay, what about that ligand-independent repression? Unliganded receptor turning genes off.
Right. Ligand-independent gene repression. Remember, many non-steroid receptors, like TRR, VDR, are bound to their HREs even without hormone. In this unliganded state, they actively repress transcription.
Why? What's the advantage?
It dramatically increases the dynamic range of hormone action. If the baseline is actively repressed, say 10-fold lower than basal, and the hormone then activates 10-fold, you get a 100-fold change from repressed to activated.
And it explains why hormone deficiency can be worse than receptor loss sometimes.
Exactly. In hypothyroidism, the unligated thyroid receptor is actively repressing target genes throughout the body, which can be more detrimental than simply lacking the receptor altogether, where that repression is absent.
How does this repression work molecularly? Recruiting corepressors.
It's the molecular mirror image of activation. The unligated receptor conformation is specifically recognized by negatively acting coregulators called corepressors, primarily NT-HOR and SMRT, also known as NCR2.
They bind to the unliganded receptor.
Yes, they use a specific sequence motif, the corner box, to bind to a hydrophobic pocket on the unliganded LBD. That critical H12 helix in its agonist-bound position actually blocks corepressor binding. So H12's position acts as the switch between corepressor and coactivator recruitment.
What do corepressors do? Recruit HDACs.
Primarily, yes. Corepressors themselves don't usually have enzymatic activity, but they recruit histone deacetylases, HDACs, to the target gene.
Removing acetyl tags, compacting chromatin, silencing the gene.
Precisely. HDACs reverse the action of HATs, leading to histone deacetylation, chromatin compaction, and transcriptional repression. HDAC3 is a key HDAC recruited by ENCORE SMRT and is vital for normal physiology. Corepressors also directly interfere with the general transcription machinery.
Okay, and the third mode, ligand-dependent negative regulation, transrepression.
Yes, ligand-dependent negative regulation, transrepression. Hormone binds and the target gene gets turned off. This is crucial for many hormonal effects, like glucocorticoids suppressing inflammatory genes.
Mechanisms less clear here.
Less universally understood, likely multiple mechanisms. Sometimes receptors might bind negative HREs where the logic is flipped. More commonly, it might involve the nuclear receptor binding to and inhibiting other transcription factors like NF-key or AAP1 without binding DNA itself, perhaps by competing for essential coactivators. Or it could be indirect, activating a gene that encodes a repressor which then shuts down the target gene.
What about those other domains, AF1 and the AB domain?
Right. The N-terminal AD domain and its AF1 activity. It's ligand-independent, often contributes to the overall strength of activation, and its activity can be regulated by phosphorylation. Its importance varies, often more significant for steroid receptors, and it can mediate tissue-specific effects or interactions with specific co-regulators.
And the F domain? Still elusive.
Still largely enigmatic, yes.
How do these nuclear receptor pathways interact with signals coming from the cell surface? Crosstalk.
Significant crosstalk. Signals originating from cell surface receptors, like growth factors or cytokines, often activate kinase cascades like MAPK or PI3K-ACT pathways. These kinases can then phosphorylate the nuclear receptors themselves.
Phosphorylation changing nuclear receptor function.
Yes, phosphorylation can alter the receptor's DNA binding, ligand binding affinities, subcellular localization, or its interactions with coregulators, effectively integrating signals from different pathways. Coregulators themselves are also targets of phosphorylation, modulating their activity or stability.
How do receptor antagonists work at the molecular level? Blocking hormone binding isn't the whole story.
They compete for the ligand binding pocket, yes, but crucially, the conformation induced by an antagonist is different from that induced by an agonist. Specifically, that critical H12 helix often adopts a position that physically blocks the coactivator binding site.
So it prevents the on signal from docking.
Exactly. It prevents coactivator recruitment. Furthermore, this antagonist-induced conformation often favors the recruitment of corepressors, actively shutting the gene down.
Let's revisit tissue selectivity. How can a hormone have different effects in different tissues via the same receptor?
Multiple layers contribute. Differential expression of the receptor itself, obviously. Regulation of intracellular ligand levels through local synthesis or metabolism, as we discussed. Post-translational modifications like phosphorylation, altering receptor function tissue specifically. And crucially, the chromatin landscape defined by those pioneer factors determines which HREs are actually accessible for the receptor to bind in a given cell type.
This leads to the concept of selective receptor modulators, SERMs, like tamoxifen.
Exactly. SERMs are fascinating compounds. Tamoxifen, for example, acts as an estrogen receptor antagonist in breast tissue, blocking AF2-mediated coactivator recruitment, but as a partial agonist in bone or uterine tissue.
How could it be both?
It's thought that its partial agonist activity relies on the receptor's AF1 function, which might be more active or interact with different coregulators in those specific tissues, while the AF2 function remains blocked. This tissue-specific mix of agonist-antagonist activity gives SERMs, like tamoxifen, their great therapeutic utility.
Are there variant receptors, shorter forms?
Yes. Alternative splicing of the receptor gene can produce variant proteins, often lacking the LBD, meaning they don't bind the hormone, even cytokine TRR. Their roles are complex, possibly acting as dominant negative inhibitors of the full-length receptor. Other variants might lack a DBD, but still influence signaling.
Okay, now for the really paradigm-shifting part. Non-genomic actions of nuclear receptor ligands. Actions outside the nucleus. Fast actions.
Yes, this is where the old rules really break down. There's compelling evidence now that traditional nuclear receptor ligands, thyroid hormone, estrogen, progesterone, androgens, vitamin D, others can elicit biological responses within seconds to minutes. Far too rapid to be explained by changes in gene transcription.
Where are these actions happening? Cell membrane.
Often initiated at or near the cell membrane. It seems that a fraction of these nuclear receptors can actually localize to the plasma membrane or cytoplasm and interact directly with classic signaling components like G proteins or tyrosine kinases like SRC.
So the receptor acts like a scaffold at the membrane.
In many cases, yes. They form signalosome complexes. Ligand binding to this membrane-associated receptor triggers rapid activation of downstream kinase cascades, SRC, URI-NPK, PI3-CACT pathways. Importantly, the nuclear receptors aren't kinases themselves. They mediate the signal by recruiting and organizing actual kinases. For example, the thyroid receptor, TR, has motifs that allow it to bind the Stogger-C family kinase, LIN, and the PA-FIDI regulatory subunit of PI3K. Hormone binding causes a conformational change that releases active heat 85, kicking off the PI3-CACT pathway.
And these non-genomic actions can interact with the genomic ones.
They absolutely can, often cooperatively. For instance, kinases activated by the non-genomic pathway can phosphorylate the nuclear pool of the receptor, modulating its transcriptional activity. It's an integrated system.
Finally, like other receptors, mutations here cause disease. Disease is caused by mutations in nuclear hormone receptors.
Yes, though naturally occurring mutations causing complete hormone resistance syndromes are generally rare.
Inheritance patterns vary. Dominant versus recessive.
They do. If the mutant receptor interferes with the function of the normal receptor produced from the other allele, dominant negative effect, then inheritance is typically dominant. This is often seen in resistance to thyroid hormone, RTH and RTH, or PPARO ligands. LBD mutations that prevent ligand binding but allow DNA binding often trap the receptor in a corepressor-bound repressive state.
And recessive inheritance.
Recessive inheritance occurs if the mutation causes a complete loss of function, e.g. deletion, inability to bind DNA or ligand, no protein made, and one normal copy is sufficient for largely normal function. Hereditary vitamin D resistant rickets due to VDR mutations is often recessive.
And X-linked.
X-linked inheritance occurs for receptors encoded on the X chromosome, like the androgen receptor mutations, causing various forms of androgen insensitivity syndrome.
Wow, what an incredible deep dive from the vast communication networks that span your entire body to the precise molecular dance happening within each cell. We've truly unpacked how hormones orchestrate your very existence. It really is a testament to the elegant and precise biological systems at play.
Absolutely. Understanding these multi-layered cascades, the specific proteins involved, the precise choreography of activation, deactivation, feedback loops, it truly brings this field to life. It's a profound example of how finely tuned our bodies are, managing this orchestra of communication at a molecular level with just exquisite control.
So what does this all mean for you, our dedicated learner? This deep dive provides not just isolated facts, but hopefully a robust framework for understanding how breakthroughs in medicine, from treating diabetes to tackling certain cancers, are really rooted in mastering these fundamental principles. It's the difference really between knowing a hormone does something and truly grasping how it performs that essential function at the molecular level.
And as you reflect on this, maybe consider how these intricate, multi-layered signaling systems, they don't just maintain our daily physiological balance, they also offer incredible blueprints for future therapeutic innovations. Imagine being able to fine-tune cellular responses with unprecedented precision, moving beyond just switching on or switching off to truly, orchestrating complex biological outcomes based on this deep understanding. The possibilities are truly limitless.